For anyone who's joining online, our presenter today is Rance Nault from Michigan State University. He leads the DMAC at their Superfund Center. He's in the Department of Pharmacology and Toxicology, and he's a previous trainee at the Michigan State Superfund Center. So I'll just let him take over and explain his talk.
Yeah, absolutely. So is it okay if I'm sitting down? Because I know I'm going to move and I'm going to walk into this. So I'll sit down, but try to stay in everyone's view here. But yeah, I'm excited to come here and talk to you about this. And it's also really exciting to kind of learn what everyone here has been doing with data management and seeing what the DMAC is doing here. It's really exceptional work and hearing about how everyone's thinking about this here. I'm going to talk about what we're doing at the MSU Superfund DMAC. And specifically, I'm going to be talking about this tool, database, repository that we're calling ToxDataCommons, really focused on metadata sharing. And as I talk about this, kind of the main takeaway is like, this is a solution that MSU Superfund is working on that we think is going to help, not just us at MSU, but potentially help people outside of MSU, other centers, groups like STEEP. But also just a lot of the principles that we're applying here, I think could be something that's, very valuable, even if it's not specifically through ToxDataCommons. You're welcome to interrupt me at any time, ask questions, feel free. There you go. Okay. Of course, I added the Halloween theme because it's Halloween. So why care about data sharing, right? So we've talked already a little bit about this, but we generate huge amounts of data. Even things that we don't really think as like kind of the main piece of data, like what was the body weight of the mice at different times or how much food they consumed. And I come from a lot of mouse work, right? So I'll often refer to those kinds of things. This is all super useful data. Sometimes we just don't quite realize what kind of impact it can have. But we can use that data to do a lot of things. So for example, dose extrapolation for environmental sampling, right? So what should we be dosing mice with to be relevant to what our environmental exposure is? And part of that is modeling as well. What is the difference in kinetics between the mice and humans? My lab, we work a lot with gene expression, big data, omics data, done a little bit of metabolomics, mostly genomics that we do. But when we're looking at different chemicals, so we work mostly with dioxin at the MSU Superfund Center, just because it was a big part of like Dow Chemical Company released a lot of that in some of the rivers. So it was a huge concern specifically for Michigan. But even when we're looking at different types of PFAS, different dioxin-like compounds, they all have different modes of action, mechanisms. Can we start to understand what those are by comparing it to a lot of other public data, right? We've got people here working with in vitro, right, for modeling. And how do we extrapolate that to not only the animal work that we do, but to human safety, human health as well? All of these are data sets that are of value, and not one lab can do all of it, right? So we rely on the scientific community to help us answer these questions. So, and that data is out there. But we do need a lot of high quality data to complete the picture. And so just like this pumpkin, we need more data. A few other reasons why to care about data sharing. So one is also reproducibility as well, right? For a long time, we've been talking about this reproducibility crisis in science. A lot of the time, we just don't have the data to be able to reproduce it. We don't know some of the variables that were part of that. So can we get to the same answer using the same data sets? And then we sort of talked about a little bit earlier as well. Do the same study designs produce the same outcomes? And when they don't produce the same outcomes, can we understand why that's happening using the metadata? Was one strain of mouse used versus another? Someone may have just said we use mice, but mice are genetically different strains. And so is that part of why it's not reproducing, right? We can also think about validation. So NIH, basically funders are really talking about new approaches and methodologies, right? Getting away from in vivo studies, animal studies, and more into cell culture, computational methods. But how do we know that those results are correct? And we want to sort of benchmark against either in vivo studies or even human use, right? Or human studies. And so having those data to benchmark against is really helpful instead of having to regenerate them as well, because there's huge history of data that we have out there that could be very useful for benchmarking these studies, but we can't because they're not really accessible, right? Can we build statistical mathematical models to capture the differences between these models, right? So extrapolating from cell culture to mouse to rat to human. And then lastly, probably the most exciting one is discovery, right? If we put all these data together, can we start to learn new things about a new chemical, start to predict different mechanisms, things like that. And we can start to use, methods like artificial intelligence to start gaining new knowledge as well. So I'm going to talk a little bit about a very kind of selfish reason why I care about data sharing. And there's a lot of value beyond this, but for my own lab, for example, we're looking to answer questions about dioxin toxicity. And with the advent of a lot of AI approaches, we asked, well, can we start applying AI methods with what we have out there already? So we came up with this research question is, can we characterize changes in metabolism from environmental toxicants by looking at metabolism as a series of connected networks, right? So individual metabolic reactions linked together, they make pathways, those pathways make a whole complicated network of reactions, pathways, and things like that. And we can normally look at this as just sort of individual little pathways, right? We can see, is glycolysis enriched in our data set, or is amino acid metabolism changed? But we know that these things are all talking to each other. They're all interconnected in this really complex web. But AI has the possibility to start looking at this in a much more complicated way as well. So a network, right, as graph neural network is a AI method, and specifically for networks, which is metabolism, is a great representation of this. And I won't go into too much details about this work, but it's the way that we sort of work this in is that we take the metabolic network and we take a lot of gene expression data. That's information about the reaction of the enzymes. So we learn about tissue-specific metabolism, and then we add exposure data, and then we determine whether certain exposures change metabolism in different tissues. That's the overall goal from using this graph neural network method. And as you can see, each of these steps, they just need tons of data, right? We need to have data to train our models with, to validate our models with. And again, this is where data sharing has a huge impact. And gene expression has a really great history of data sharing. So it's probably one of the first data types where publishers started requiring that people submit their data to the gene expression omnibus. And so we have a lot of gene expression data that's publicly available. But as you'll see as I go through this, it's still not perfect, right? There's still things that we need to address. Okay, so first thing was we need to train this method, right? We need to train this neural network. So what we did is we went and basically downloaded pretty much any mouse gene expression data that we could find. We worked with this database called Recount3, where they took a lot of mouse data sets, mouse gene expression data sets. They analyzed it through a common workflow. So every data was processed through the same workflow. So those counts that came out were all consistent. And they all took the data from Geo to create this. So we started with 225,558 samples. Great, right? This is awesome. And we were really excited. I mean, we've got lots of data that we can work with. That's good for AI, right? More data, better we can do. But then we had to start looking at, can we actually tell what these samples came from? What are they telling us? Are they actually relevant for the model that we're training? So we're training a model to learn about mouse metabolism. And so we had to start excluding genetically modified mice, right? Because they're going to be a little bit odd. We excluded any data set that was from developmental stages, because that metabolism is going to be different than an adult or mature mouse. Also had to exclude all the in vitro studies, because that's just not, you know, a hepatocyte is not the same thing as a liver. And so we had to filter for all those things. So obviously we lost quite a bit of data when we added those filters. And then the next thing we wanted to do is say, well, do we at least have enough information from what's remaining to start training this model? Do we even know the basic things like what strain was this from? Because we know different strains are going to be, their metabolism is going to be different. Can we tell from which sex these samples came from? Were they treated? Did they receive any treatment? Because if you recall my previous slide, our first goal was to determine tissue-specific metabolism. And if we're starting to add in perturbed samples, then we are kind of learning from abnormal samples. So we want to make sure that we filter for things that are normal as well. So we added all those filters. And after all of this, we end up with only 7,900 samples. And I wish I could go and break it down specifically, like for why each one was excluded. But I can tell you right now that it took us about 2 to 3 months to just figure out what we need to include or not, just because the format was so inconsistent. Someone called it strain, source, mouse, you know, all those kinds of things. And we had to try to figure out how do we even start filtering. And so a lot of this was done manually as well. And that's the other thing that's kind of hidden here is that we lost a lot of samples. Probably a lot of those we lost as well because we just couldn't find the right information, right? And so that's where things like templates and things like that come into help a lot as well. But we had 7,000 samples, which is actually not too bad for training a graph neural network. But the toxic perturbation samples were even more challenging. So I first want to talk about 7,900 samples. So what you want to look in this type of graph, where this is my mouse? Oh, my mouse is showing up. Okay. Right here. It's showing up right now. Okay. So essentially anything above here is performing really well. It has high sensitivity, high specificity. Anything that would be down here would be poor performance for predicting which tissue a sample came from based on this graph neural network of metabolic pathways. So we're seeing that even with about 3.5% of the samples that we kept, we have really great performance for predicting which tissue these came from. So it was enough data to learn and to build this this model. But like I mentioned, this became a lot more challenging when we started working with samples of exposure. And first of all, you could probably imagine there's just fewer in general, right? So that is one challenge. But the main one was we just couldn't find the metadata. There was a huge underreporting. And this was something we were sort of aware beforehand. So this table right here, we went into GEO, the Gene Expression Omnibus repository. We pulled out every single metadata worksheet that they had or spreadsheet that they had. Again, it took a lot of manual sort of renaming some of the columns. You know, people use different terms for the same thing. So we had to put everything kind of on standard terms. And this is really small. Don't need to read every one of them. But the main thing is this. So this dashed line here is 25%. And you can see that some things are relatively well reported. So things like strain are well reported. And part of that is Geo was pretty good at saying this is a suggestion that we should include, right? Strain, sex was another one, because no matter what field you're in, that's probably something you're reporting for an in vivo study. But once you get to sort of toxicology studies, environmental health, right, exposures, now we're well below 25% of even just reporting dose. How can you work with toxicology data without dose, right? You don't know what you're predicting. It could be a high dose, it could be below any level that actually causes something. The other one I highlighted is administration route, whether they were gavaged, IP, inhaled. We have no idea, but that affects the kinetics, that affects the tissue levels. So we're missing so much data. And so it was really difficult to work with this. So another Halloween theme thing. So how do we unravel this data management, right? How do we fix this problem? So this is beyond just the sort of selfish need, right? So I talked about using AI and we wanted to develop this model. But there's just broader advantages as well for doing this. So this is a graph of how much money the NIH and other funding agencies have been given, and it's billions of dollars for research to generate data. But how many times are those data sets duplicates of each other because they're not being shared, right? How can we maximize that return on investment from generating these data? So one way I like to talk about this is we do a lot of single cell RNA sequencing and spatial transcriptomics. These are really expensive studies, right? Spend 20, 30,000 on a single study. And if another group proposes the same thing, that's another 30,000 we're spending for the same thing. And another group's another 30,000. While if they could use it, they saved with that amount of money if they can reuse our data sets. But the main thing I want to also note is that the goal is not to just not reduce studies, because reproducibility is still important. So sometimes they need to be reproduced. But let's make sure we have that data so that it can be compared to, all right, so that we can really add that value. And then that money can be spent to do something else, right? That essentially led to these big initiatives. So you've probably heard about FAIR a few times: findable, accessible, interoperable, reusable. So FAIR principles now are kind of central to NIH data management policies. And this is a quote from one of the documents they put out. So they say data management sharing practices consistent with the FAIR data principles. So they aim to require data sharing of all research data, right? So that's a huge goal for them. And that is, they state exactly, regardless of whether the data was used to support scholarly publication, right? So what is all the data that we don't publish that is of value? Because there is value there as well, right? Negative results, things that don't make it publication. How do we share those as well? And those are a little bit more challenging because you still want it to be good quality data. And then Superfund programs. So Superfund, I think we're kind of leaders in sort of implementing these data management principles when they sort of require these DMACs as part of the Superfund research centers. And the goal of the DMACs, they say, was to support the management and integration of data sets. So all of our groups that are doing similar data sets but working on different problems, how do we integrate these, right? Because we've already talked a little bit about earlier today about how some data sets can be used for modeling, for those kinds of things, right? And then the FAIR data principles. So Joanna shared some good links about FAIR, but this is kind of the outline of what they want for FAIR. And it's a lot of data, a lot of information. I won't go one by one, but as I talk about ToxDataCommons, I'll sort of point how it addresses some of these things. But there are some expectations for data management and sharing. But the thing I also kind of like to emphasize is that FAIR principles were never put out as requirements, right? They were set out as guiding principles, and it specifically says guiding principles. Because how do you do these things, right? There's so many different ways to do it, and it's really hard to enforce or force someone to do that, especially when we don't have the infrastructure. But these are concepts that we want to apply for data management. And I think these are really important ones. The main thing that, when we were looking at FAIR and we were sort of implementing our DMAC for the first Superfund cycle, was looking at what are these FAIR principles. Again, I won't read through every one of them, but I highlighted something specific, which is, as you can see, in almost every one of these items, they talk about metadata. And as I talked about as well, one of the main things we were missing from those studies when we were trying to build the neural network was we don't have the metadata, right, to be able to reuse it. The data was there, right? We have 200,000 samples. We just don't know anything about those samples, or very little about those samples. And so our center sort of looked at this and said, well, we really should start thinking about addressing the metadata question. So how do we basically stop going from 200,000 to 7,000? How do we fix specifically this problem, which is really concerning to us, right? This very low reporting of dose and administration routes. Now, I talked about kind of how bad the state is, right? How, it looks kind of bleak in terms of what we've done in the past. But the thing I also want to highlight is that we're actually kind of good at reporting metadata. It's just how we're reporting it. So in that study where we pulled all those terms from Geo, we pulled the 3 studies that did the worst. They reported nothing in the Geo metadata. And we actually pulled the publication that came with it. So you can see they were in the metadata in Geo, they reported nothing or 1 item. But when we actually looked at the paper, I mean, 38 of the terms that we were looking for were in there, 45 in this other study. This one was a little bit lower. So they were reporting it. We just couldn't really access it, right? So it's already something as scientists that we do. And we talked about this morning, right? All this stuff that you collect in your Excel sheets and your notebooks and all those things, it's there. Main question is, how do we get it to the next researcher in a way that's useful? So yeah, as much as I talk about it, it's kind of bleak that it's really hard to do. It's there. We've always thought about this. It's not readable by machines. That's the main challenge. All right, so how do we get here? So when we started looking at this, we're kind of thinking, well, how come it's kind of so hard to get to this point? And really to us, it was looking at what's available. And we kind of came to the conclusion that it's the tools, the infrastructure, and how accessible those things are. So we didn't touch on this a whole lot today, but how many of you are familiar with reporting standards for your data types? Not a whole lot, right? So where are the reporting standards, right? How do you find them? Are there reporting standards? When you find them, how do you actually apply them? So if someone gave you a list of reporting standards, how would you implement it in your lab, in your research? Right? Would it just be an Excel spreadsheet, right? Or would it be another way? How do you make sure that they're applied correctly? Because you can interpret those standards one way or another, right? How do you make sure that it's actually fulfilling those standards? And now you have those standards, you have everything. What do you do with it at that point, right? What happens next? And those were all things that we found didn't really have great resources. And I will say when we started the DMAC, I think the state was a lot harder to find a lot of these things compared to today. So there's definitely been progress. So essentially what we're talking about is, how do we go from the lab to FAIR? And this is the way I think about it mostly. So I've done all my training in the lab, right? I'm a lab scientist. I do computational stuff as well, but I really started working at the bench, right? Doing these in vivo studies, in vitro studies, all of this, and thinking about, okay, well, I've done this. How do I get to the next step? And by, applying these computational methods, sort of learning oh, I should have had this because it would have made it more useful and those kinds of things. And we started kind of at the start of microarrays, RNA-seq, when all the standards for gene expression data was coming into play. And I think gene expression data set a good sort of framework foundation for some of this, but there's still good ways to go as well. So we looked at the steps between this, right? We said, okay, well, we're at the lab. What we need to do is we need to know what standards we need to meet. We need to have a method to apply them. We then need to collect the metadata following those standards. And then we need to put it somewhere. It seems pretty simple. It's probably oversimplifying it a little bit, but I think it's a good kind of overall concept. And then we looked at, okay, what do we have available for each of these steps? So identifying standards. Has anyone been to fairsharing.org? One couple, yeah. So fairsharing.org, it's developed by the Elixir group in Europe, is a phenomenal resource for identifying standards. They've kind of become one of the place where these go. Now, that's not perfect. There's a lot of standards that are in other places, which is why I put PubMed as well. And this is how you can identify them. Not quite addressing the challenge of how do you determine that it's the right one, it's the best one. That is still a challenge. this is where experts in your field, right, really come into play, right? You are experts in your own data type that you're generating in your own research. And if you find a standard that's asking for something that would never be relevant, is it the right standard? Or if your standard doesn't say, well, you should collect dose, but clearly dose is a thing that we should collect. Well, clearly it's not the right standard, but maybe it's something that can be adopted. But there's some challenges there, but these are a couple of really good resources for finding standards. All right, so now that we have some standards, how do we implement them? And there were some tools around that were already available. So this group, ISA, Investigation Study Assay, also from the Elixir group that does fair sharing. Again, a lot of this came from gene expression, from the gene expression world, microarrays and RNA sequencing. And they had sort of implemented some standards for sequencing already. And so they developed some tools for collecting that. Data Harmonizer is another tool from the University of Vancouver. That's a lot more customizable. So it's less of like standards from these places as well. And CEDAR, which is the Center for, I forgot what it is. But anyways, it's a workbench for collecting metadata as well, more in a form type format. But these are a few resources for implementing them and also collecting them as well. So it kind of mix both together. And then finally, where do you put this data? And there's a lot of places, right? There's a surprising number of places to put these data. So Gene Expression Omnibus is standard for gene expression data. SEBS is a place for toxicology data. Now, the submission to SEBS is really, really hard. Metabolomics Workbench. So I've heard a lot of LCMS, a lot of mass spec. Metabolomics Workbench might be something worth looking into. It is a little bit less about, it's called Metabolomics Workbench, but it doesn't check whether it's metabolites or environmental contaminants or anything like that. And then Data Dryad, Figshare, Harvard Dataverse. So there's a lot of sources for these, right? But also there's a lot of gaps in these, right? And they're all pretty different. So how do we find the right place to put these? And when we were looking at this, we were finding that there's just a lot of disconnects along the way, right? There's a lot of, this one isn't well integrated with that one. That one's not well integrated with this one. It was really hard to work through everything. And so we set out to develop a few tools that hopefully would be useful. First one is called Tox Reporting Standard Catalog, SheetMATE, which is our metadata collection. And mostly what I'll focus on today is ToxDataCommons, and how we sort of came to that point and implemented that. Okay, so what is ToxDataCommons? Our ToxDataCommons is live right now. We're making some updates, so there's not a whole lot in there right now, but toxdatacommons.com is where it's at. So this is intended to be a fair resource. And really what we're focused on is collecting and sharing and helping discover AI-ready data sets. So again, being a little bit selfish, we want it to be AI-ready because we'd love to be able to use AI tools on this, as well as just sharing the data for whoever wants to use it. And the main thing is that we're not looking to replace what's out there. And I'll touch on this a little bit, but what we also do is we connect some of these to these sort of standard repositories, right? We're not going to serve as the repository for gene expression data. Geo does this already very, very well, but we will help collect the metadata for those studies because Geo does that okay, somewhat poorly, right? I mean, it's all based on this data model, which I'll talk about. Okay. I don't know if there's any questions so far. Perfect. So ToxDataCommons, its foundation is the data model. Really, the data model, I think, is what gives this a lot of its strength. And it's what we spent the most time preparing. It's also, what I think gives it value, but also makes it challenging to expand to other data types, but we're working on that as well. But what we did is we took a lot from the lessons from the past. So I talked about ISA Creator, ISA tools. which stood for Investigation Study Assay Tools, again, came out of all the gene expression work. And this was a consortium that essentially looked at if we wanted people to just share any type of data, like what is the most convenient structure that we could use for like saying like we need these kinds of things. And they said, well, we should get investigation details. And that's like funding, which lab it was in, publications associated with it, just overall details, right? And then study would be the next level, right? So subjects, mouse, humans, samples, right? So kind of upper level, what goes into the next step. And then finally, assay, right? So what are you measuring? So is it gene expression data? Is it imaging? Is it mass spectrometry? All those kinds of things. So we sort of built it on that framework we have different levels. So investigation, study, assay. And for us, the advantage that we see here is that when we have a new data type, it can still connect to that same root node subject, but we can now add clinical chemistry. We've done blood pressure measurements, those kinds of things, right? But they're just different assays that get added along the way. So, but really we focus on the metadata about, overall experimental design. So mice, humans, geographical location for sampling, right? So what I talk about here, a lot of it is for in vivo experiments. Again, that's where we kind of emphasize first, but we're also working with people that do remediation work and things like that. So thinking about these other components as well. And this is all based on standards that we developed, which is called the minimum information about animal toxicology experiments. And this was, again, a super fund effort with MSU, University of Kentucky, University of Louisville, and University of Iowa, where we sort of looked at those reporting standards. We said, well, these are some gaps that we're seeing in those standards for toxicology. These are some of the good things. And since we all do in vivo work, this is a consensus that we kind of came with for what we should have in these. And then each of these assay steps, we also, as much as possible, implement reporting standards that are associated with those. Now, there's some that we can't find any. So, for example, like reporting of body weights for animals. We really couldn't find anything about that. But for gene expression, we have the minimum information for sequencing experiments. For imaging, it's the recommended metadata for biological imaging. CIMR, which is the core information for metabolomics research standard for mass spectrometry. And then we also kind of connect these to these known repositories for it, essentially giving it its strength for being able to then also go to those repositories as well. In that data model, right, it's not just saying these are the nodes that we want, but it has what is the information in those nodes that we need. So you can see, for example, that we have a list of what are the terms that we should have. So sex is clearly one that we should report for animal studies. Strain is also something we should clearly report. And we have a pretty extensive list of these things. So it can get pretty complicated, right? But it gives you an overview of kind of what are the minimum expectations, because these are all built based on these minimum requirement or reporting standards for these studies. And so we list all these things. What we also do is we sort of enforce the use of certain terms, some ontologies or controlled vocabulary. So one key example that I always like to point out is that, for strain, for example, we'll only allow people to upload specific strains, has to follow this exact name, because we found when we were doing that analysis of the geodata, someone had reported Babel C instead of bulb C for a mouse strain, and we couldn't detect that easily, right? We have to also look for typos. So that's going to be a nightmare, right? So how do we prevent those things as well? And that is something to think a lot about when you work with like spreadsheets, right? Because typos are really easy to incorporate. So how do you reduce the chance of that happening? How do you make sure that you're using the right term, a consistent term, and not accidentally introducing a typo that you did everything right, but I can't find it because 2 letters were switched, right? So this is the overall picture of what that data model looks like. So it's pretty complicated. There's a lot of things in here, but you can sort of see the investigation level, the study level down here, and these are all the assays. And as part of the assays, we have both the raw data and the analyzed data at the bottom as well, some data analysis steps as well. We haven't quite implemented that all the way through. So we're working on, through some things in here. And so our data model is evolving, and we're looking for users to help prioritize what we need to put in here, right? So we have a lot of in vivo users, and so that's relatively simple, straightforward for us to do because we do that kind of work. So we talked about this already with Joanna, but addition of dam-sire relationships for reproductive studies. So that is, for example, something we hadn't thought about when we first did this. But clearly it's something that you do here at Steep, right? So this should be something we consider as incorporating into the model to address. But what's interesting is there was no reporting standard about that, right? So how do we do that? We're also working on integrating a environmental sampling module because we also do some remediation of activated carbon, one of our engineering projects, using activated carbon to sequester dioxin. So how do we measure the dioxin availability in those activated carbons? And once we gavage mice with those activated carbons with dioxin, what is the bioavailability at that time as well? So it all connects together, which is the goal of Superfund, right? How do you integrate those data sets? And we're also working on a community survey module as well, but that one, it's a little bit harder because it's a little outside of what it is, but we're looking at ways to integrate that as well. So the other really nice thing about having that data model and having this standardized sort of structured metadata collection is that we can now explore this in a very structured manner, right? Before we were working with spreadsheets trying to find the right terms, but through, there's an exploration node on ToxDataCommons. But because everything's consistent, we can then just start selecting. We know that this is a category we can search by. We know the strain is a category we can search by. We can count how many are in each of those. And so we can do those kinds of filters and find the right data sets without having to do really complicated. So for example, for TCD, we may have to do TCD, or someone may have used dioxin, or they may have used the full chemical name. Someone may have called it liver or hepatic or hepatocyte. So, we need to search all these things. They're all different ways to say the same thing. But with the standardized method, then we can better find this. And that's a core aspect of FAIR, right? Findability was a key thing here. And from that, we can build cohorts of data sets. We can pull all the imaging data. So this is, I gave this as an example, it's just a bunch of test data as we were developing it. But for example, we would pull all the imaging data from a study, and we have a DOI, or a persistent ID, I should call it, for each of these. And each of these can be accessed, downloaded as a batch, right? So if you're looking for all the environmental sampling for a specific region or a specific area, you could pull all those out, download all those data sets, and start processing it. And it's all standardized. And so you can work on the data more quickly rather than like what we did for our graph neural network, which is spend months even just preparing and finding and all the data and everything. So this seems great. I mean, when we kind of walked through ToxDataCommons, we were kind of looking at what do we address in FAIR. We kind of went through each one. We're finding that I think we're addressing up pretty much every aspect of FAIR, some better than others. But we are kind of focusing on these, right? And how do we address each of these points? Because, for example, community standards, we're implementing community standards. Usage license, we are working with permissions, like who can use what and when, right? So if it's data that's not published yet, we keep it behind a password so nobody can access it, right? And so it kind of, in a way, addresses some of the how existing resources are pretty much siloed, right? What we're doing is we're putting it all together. So our data sets may end up in Geo or Metabolomics Workbench, and once they're in each of those, you can't connect them, right? Unless you're very good at using the same IDs and then somehow telling people that they're both at two different places. But here, we can connect through metadata, point to those locations, and find things that are also from the same samples. And I should mention that ToxDataCommons, so we developed like the data model and things like that. the framework that it's on. It's the Gen 3 Commons. And I can talk more about that. That's more technical, but they are pretty well established in this field for managing data. So problem solved, right? Everything's good. No. So how do we get this into ToxDataCommons, right? Because I talked about how great ToxDataCommons is and how it helps structure everything, but we haven't really talked about how we can even get things in there. And so our next step was kind of looking at how do we make that collection accessible. So there's a lot of efforts that I sort of briefly mentioned about trying to do templatization. And we kind of talked about it this morning as well, right? Everyone's got templates in their labs that they're using. But how do we do this in a more robust manner, right? ISA tools was one, Data Harmonizer, Cedar. But who creates those templates? Who has access to those templates? Do we use web forms or spreadsheets? These are all questions, right? When we talk about templatizing metadata collection. And when we look at this, we kind of think, okay, well, who creates the templates? It should really be the domain experts, right? People that know this field, that understand it. And it should be pretty much based on those reporting standards. Who should have access to those templates? That should be pretty much everyone, right? You want people to collect things in the same way. So everyone should be able to access that. The main thing that was interesting to us, so Cedar is pretty well established for metadata collection. They're very good for especially more clinical, more human-based stuff. They use a form format. And we found really quickly that for us, this form format just doesn't work. We work with 100 mice. You don't want to do one at a time. And it sounds like even talking this morning, even here, spreadsheets is kind of what people are familiar with and like to work with. And then the other thing that we really want these forms to be is machine actionable, which basically means that it doesn't have to be a human that, creates the template or allows you to populate that template. You want it to be readable by a computer because that makes it a lot easier to implement with different tools. So we set out to kind of build another community resource. And again, we're basing this on standards that we find in fair sharing. So I list a few other reporting standards here. And we worked on sort of making them machine actionable, right? Readable by machine. So using a LinkML, which is a way to format these standards, saying what do we need. And this is available on our GitHub. You're welcome to take a look at that. And then this input, we have to make some small changes, but it's the input for this data model. This is what's used to essentially say to ToxDataCommons, this is how you expect the metadata to be organized, structured, and all that. It can also be used to do templates through Data Harmonizer and Cedar. We haven't tried Cedar, but we know like the formats are compatible. We used to use Data Harmonizer. We ran into a lot of challenges with that. So we ended up doing SheetMATE, which I'll talk a little bit more about. So SheetMATE, basically driven by user experience. So we originally started using ISA Creator, which actually was a really great tool. It got a little hard to use, but it basically got retired. They no longer wanted to maintain it quite a while ago. It was machine actionable. It was just not the most convenient machine actionable format. And then we looked at Data Harmonizer. It builds from LinkML, but it had a lot of limited functionality compared to what you're used to on a spreadsheet. So like we were trying to use it like we do Excel, and there's a bunch of things we could do, and it just got frustrating, right? Because you're like, I could do this in Excel. Why can't I do it here? Cedar was a web form. And so none of the tool, and then the last thing is that none of these tools we could directly connect to our ToxDataCommons. And when we're thinking about this is how do we reduce the barriers to uploading data, uploading metadata? And for us, it was, we want something where people can put in metadata, hit submit, and it's done, right? You don't have to do a bunch of other things with it. So we did SheetMATE to essentially close the loop. And this is basically just a tool based on Google Sheets. So everyone's worked with Google Sheets, pretty much Excel, right? And what it does is essentially it goes to ToxDataCommons. It asks, okay, what is the standard? What is the requirements for this part of the metadata model or the data model? It builds a template off of that, tells you, what is expected in each of these. When there's certain rules, like you have to use a specific vocabulary, you have to use a number, you have to use a date. It pulls that information in directly into that. So that template is already built, always based on the most recent version of the data model. And we keep track of which data model as well as we do this. And then the next step is you enter data like you're used to, right? And then you hit submit, does a few validation. We're working on a few more. It's not perfect, right? There's some validation that we want to add. And then it submits that directly to ToxDataCommons. And so it allows you to sort of build on to each node and keep populating that. So you can start from the first node, walk away, come back the next day, do the next node. And as you start collecting your data, you can do the next one. Our SheetMATE is not quite ready to go out yet. We're fixing the fully automated step right here, but that's essentially the way that this is intended to work. So I'll just kind of wrap up here, I think, to kind of a couple examples, and they're pretty simple examples. So it's not really fancy use cases, but I think they're more simple ones are kind of a good way to show how we can use this. But one of the ways that we actually are using ToxDataCommons is actually to help support uploads to these standard repositories. I talked about Geo Metabolomics Workbench and how we can pull metadata from those, but we don't know what to expect, right? How, like, what did they call the columns, like that? But what if we have a template to upload to those that is broadly accessible? So this is an example here where we were submitting data to Metabolomics Workbench. So mass spec data, Metabolomics Workbench. I didn't plan this, but it sounds like it'll pretty relevant to everyone here, right? We were submitting metabolomics data to Metabolomics Workbench before ToxDataCommons. So coming from the history of doing this for gene expression all the time, it was something we had thought about even when we started working with this type of data as well. And these are two studies that we submitted before. So one of them has a treatment summary, it's just a big paragraph, has all the information. And Metabolomics Workbench is completely happy with that, right? We've given them what they need. Another example, we tried to do a little bit better. We broke it down into different categories. What is the treatment route, the volume, treatment vehicle, right? Specifically saying what vehicle we're using to prepare this. So you can see it's widely different, right? So when someone wants to use our data and they see this one first, they can't find this data now in this other one. Now we need to do everyone manually, right? But once we started using ToxDataCommons, these are the submissions that we're doing. Now you see the exact same elements are in here, right? Structured in the exact same way. We're using controlled nomenclature. So instead of saying what the chemical name, we use this DTX ID, because that way, if someone says it's dioxin or TCD or 2,3,7,8 tetrafluoro or whatever, it still has that ID that represents what that chemical is. So you know that you can look that up and say, I know what this is. The doses are structured in a similar way. All these other information are, and even for the vehicle, we say what the DTX ID is, right? So all of these endpoints we're standardizing by using ToxDataCommons to build that upload package for Metabolomics Workbench. So that is one example of how we use it in a very simple way, beyond just before even just the benefit of like reanalysis and things like that. Another thing too, and this is a very small project, so we're still, looking for more data sets to input, but how can we integrate data sets? So we have data from at least 3 independent studies in here. They had different study designs. And sometimes that's really hard to extract when we're just pulling, the data from Geo or something like that. But can we get some informative patterns about this chemical when we're working with this? And this is not super fancy and it's not really surprising or anything like that, but we are able to take these nodes here and pull what was the metadata associated with it? What was the sex? What was the dose that was used? And when we look at body weight, and we did some just simple analysis for matching time with dynamic time warping to compare what is the effect on body weight for each subject. Well, now we can see that these samples separate, and we can annotate with that metadata and see, for example, males at 30 micrograms per kilogram deviate quite widely from all these other samples. In fact, it tends to separate along this axis by sex and this axis by treatment duration. We can do that because we have that metadata available in a computationally accessible way, which helps a lot. And we can also then correlate that to liver weights, because we collect liver weights when we do this as well. So how do the body weights correlate? Well, we can see that this is again the males, and some of them, the body weight correlates really well with that relative liver weight. But we can start again incorporating some of that metadata. So in terms of like a research question, I mean, this is not something that surprises us at all. TCD increases liver weights. And also affects their body weight as well, especially at these higher doses. Not surprising, but it's just an example of, because that data is structured, it's a lot easier to just start doing these analyses and pull that metadata and start integrating these different aspects of the data. Okay, so just in summary, so improved data sharing has the potential to benefit, right, the whole environmental health research community. So I think sometimes we don't quite know yet what impact our data set can have. And I kind of like to say, like, share it, because someone may find a use for it. Even if a study where you're just publishing your body weight data and we don't really care about the treatment, that body weight might still be valuable. So we tackled this by trying to build the infrastructure. It was a huge undertaking. It's tough. But like I said, it's only a single piece of the puzzle. And whether it's the right piece, we'll find out, right? But I think at least it should get people thinking about how to collect metadata in a standardized way, how to make it useful for the next person that's going to use it. And we're looking for collaboration, feedback, ideas, all that. We're doing this at MSU, but we want this to be a community resource. We want this to be useful to everyone, because our goal is to advance research, to be able to just drive discovery and focus less on having to repeat studies or redo things, or kind of read through, trying to find all the metadata from papers that everyone's published. We kind of have a vision, but we're kind of looking to change it. So right now we're working mostly with Superfund. Our goal is then to extend to MSU EHS researchers and then external data. We're looking to shift that around, because clearly there's other groups that are interested and willing to start working with this as well beforehand. And we're pretty flexible in how we incorporate things, but we're hoping to show the value first. That's our main goal, right? Does it actually provide value? And we're always looking for people to work with. And so I'm just going to wrap up with some acknowledgements. So I have to highlight KG here, that's done a lot of work in sort of the computational stuff to set up the data model and just coding and all that stuff. Huge team that's been working on this. Eric Casson was the DMAC PI at first. He retired, so I took over. CTDS, that's the group that developed the Gen 3 Commons platform. That's the University of Chicago Center for Translational Data Science. Phenomenal team to work us. It's all open source, right? So, and then Crumware are the ones that are helping us do the really hard stuff with AWS, Amazon, and servers and all that stuff. So with that, I'm happy to take any questions.
Are you currently working with any other Superfund research project programs? Are they actively submitting data?
No, so we haven't. So one of the interesting things about the Superfunds is, like, everyone came up with their solution at first, right? And so it'll be interesting to see like which methods kind of end up, being adopted more widely. So we've talked to other Superfunds, but right now we're mostly MSU people that are using this.
I think my question also kind of goes into this. So if you take, for example, Olga's study data, what are the steps that you need to do to get it integrated in ToxDataCommons?
Yeah.
So I kind of understand the benefits. It's more like, how do you get there?
Yeah. And this is nice because Olga studies is a pretty good example, because it's in vivo studies, right? And what we would do is we would send you SheetMATE, the template, and you would go in and say, I want to start putting in the study data, where you would put in, this is the lab we're working in, this is the funding source, all that. You don't even need to start your experiment at that point, right? And then once you plan your experiments, and you've got, okay, I'm going to be doing a study with 20 mice, and these are going to be the identifiers that I'm going to use for them. So you'd go in and you'd say, okay, I picked the subject node. I might be able to. So this is our data model, right? So each of these nodes, you can kind of go as you progress through your study. So program project is something we assign. So we'll say this is the URI program. And essentially you start at study. And this is where you would put in, is this an in vivo study or an ex vivo study? We're working on in vitro studies as well. This is the organism we're working with. And these are all things that we can also expand as there's more. But pretty simple, right? Just a couple of terms to include here. But we, through SheetMATE, have a template for you to basically put in that information in spreadsheet format, hit submit, and it goes up here. Now, I can't show that yet because again, I said we're fixing one little piece on that. So it's been a beast to work, but essentially the idea is that it keeps telling you where where you're at, so that you know what to do from next step. And that's kind of the piece that we're working on to make it truly user friendly. But then you would start your experiment, right? And you say, okay, I've got these are the mice that I'm going to use. So these are the subjects. This is where you would give them their IDs. And you don't need to have all these dates yet. You can update it as you go as well. So that's one of the nice things as well, is you can update as you go as well. And you would put in that metadata. Was it male or female? What was the strain? Again, strain source. We would like to know where did those mice come from, right? Were they internal, Charles Rivers, Jacks, wherever? And then you can start your study, do whatever you want. And here we have a treatment, which connects to subject right here. And you wouldn't have to go step by step like this. The SheetMATE would basically say, which node do you want to populate? Do this one next, do this one next, do this one next, right? So this is just kind of a way to see the overall structure, but this is how you would add it. And so here we have some free-form treatment protocol because sometimes we can't catch everything. But we have things like what volume was added, right? Or sorry, that's how much of the chemical was added to your stock chemical to your solution, how much was administered to the mouse or rat or whatever. Units, right? Making sure we're reporting units, because that was one of the biggest nightmares for working with GOs. What unit were they using? Dose unit. The final concentration that's administered, right? Which route? And we have this massive list of routes. So we think we have pretty much everything covered, but there's things we can adjust as well. But these are all things that you would kind of know as you start your study. You even had in your template, not quite this right here, but you had the time, right? So if you look at just relative to when the lights turn on in the animal facility, and we specifically focus on, we want it relative to when lights turn on, because that's really what matters for mouse studies. So we collect that stuff as well, because that also matters a lot. And when we were doing these reporting standards, we're like, well, we really should know when we're treating them and when we're collecting these tissues as well. So yeah, the template would give you these rows as columns. It would give you these description as little notes in those columns, and you would enter your information. You could even copy from whatever spreadsheet you're using as well. And if there's a typo, it'll tell you. If it's one of the ones, like if there's a typo here, if you put just UG, right, for microgram, it'll tell you, no, you can't do this. We want one of these options. And that helps us make sure that everyone's working with the exact same thing. This is mapped to ontologies for units, so that anyone can look up, like, this is what this means specifically. So that's a kind of a small overview of that. And then you would just keep going, right? As you collect your samples, you would put in sample information. We then have aliquot. It gets a little complicated because it's sample and aliquot. That is a Gen 3 requirement, which we unfortunately couldn't really work around. But we still like the thought of using an aliquot, because you may use part of the liver, part of the serum for one thing, and part of the serum for another thing. But each of these steps ask for specific terms. And again, that template would walk you through each of those steps as you go. You could leave, say, all right, next day I'm working on samples. All right, and then this next day I'm working on the mass spec data. And so what do we need for a mass spectrometry assay? Calibration standards, what is the carrier gas, what is the column we used, chromatography instruments. And one of the things about the list that we built here is this was based on the requirements from CIMR, but also from Metabolomics Workbench. So this is exactly the term that they expect when you upload data to that database. And so that way, you know that everyone's using the same term, but also it's compatible to deposit it where it's expected to be. Because that's the other thing too, like ToxDataCommons is not the first place people will look for metabolomics or mass spec data. So we are making sure that we're meeting like what are those, if there's a database that is the standard, we're not trying to replace them. We don't want to, plus they can pay for the storage. So but yeah, that's kind of the steps to go through for submitting that.
So what if somebody used a new instrument that is not on your list yet? What do they do at that stage?
Yeah, so that would be us that would have to add it into the list, and we can do that easily. So we have a certain sets of changes that we can make that are considered breaking and non-breaking. Adding items as non-breaking, we can definitely do that. Even changing, so if someone says, well, we should stop calling it this, and we should start calling that, we can change that as well. We keep a version of the dictionary. So these are the versions that it's working on. So we're always aware of which version we're working on. So if someone had collected all their data using an older version that said it's called machine X and now it should be called machine Y, we know that it was collected with the data model X, 1, and so it's going to be machine X, and machine X is equal to machine Y in the new model. So we're trying to prevent, address those as much as possible. Because these are definitely problems that we've run into before. But adding things adding elements in these drop-down menus is really easy to do. Yeah. Yeah.
So Google Sheets or Google in general likes to change their UI and stuff kind of randomly. Does that cause problems for this?
We haven't had it long enough to see if it's going to be a problem. Hopefully not. But we don't know how to avoid that because it's more of a problem with Excel, the actual software, because the ISA group tried to do one in Excel and Excel updates and doesn't work anymore. So Google Sheets in the backend doesn't seem to change as much as long as, and it's more computational, but as long as the input is in the right format, it should still fill the spreadsheet in the right way. And we don't really work with the UI of Google Sheets a whole lot. The only thing we do is we add a drop-down item in. So there's a drop-down menu that says ToxDataCommons, submit, or populate data in with that.
And then is it being stored within Google Sheets, or is Google Sheets just kind of a bridge to get it to the accessible online?
So right now, we do directly on Google. We do both because again, we have that one little piece that connects it that's not working. So right now, the way that people are doing it is download it to your Google Sheets, then upload it to our folder. The last piece that we have it working, so that's not the piece that's not working. But when you hit submit, it goes directly to our Amazon bucket. And ToxDataCommons is essentially looking for new files, and as soon as it sees a new file, it puts it into the repository. So eventually it won't be on Sheets at all because there's some concerns about Sheets and being able to access it and things like that. Our S3 buckets tend to be a little bit more secure. So yeah. The other thing that I haven't quite highlighted as well is there's two ways that we can work with ToxDataCommons, and one way that we've seen for people where we haven't been able to address all their metadata requirements yet. We have what we call the data lake model, which is more like Harvard Dataverse, where we can just upload the files and connect it to a study, to a project. And so we can look for certain types of files as well without all the metadata. We found that to be especially helpful for people who are realizing later on we need to do this and how much work it is to put everything all at once. Like doing all those nodes at the once is a lot of work. Like we absolutely understand that it's asking a lot to do this because it takes time to enter it, take the time to put it in right and all that. So we have a method where we can also just upload the data file. That data file has a persistent ID. And then you can go in later on and add in those metadata as you have time to populate as well. So we have that mechanism working as well.
So assuming you have a workflow like you described initially, where you begin at the early stage of your research, I'm assuming that when you do finally get around to uploading your data set, there's some quality, the data has to conform to the template in some way?
Yeah, so this is a great point. And I will tell you, so we focused on metadata.
Yeah.
So we don't have very thorough checks for the data quality.
Mm-hmm.
So if you upload a file, like an image that is just empty,
Yeah.
we won't look for that.
Mm-hmm.
We don't have the mechanism to do that for every
Right.
data type. So we sort of rely on the researchers to be the experts in their field to do it right, to have those processes. We are building some. So for some more free-form inputs like body weights,
right.
we do have a couple of things like we should call a column value and a column submitter ID so it can match to the subject.
Mm-hmm.
So we're starting to build those, but those are more one by one,
yeah.
And we're focusing on the ones that we do more. But yeah, otherwise, like, yeah, we're not QCing the data quality.
Right.
Yeah. So that's definitely a limitation, but I mean, we are checking that. Actually, no, we don't do this yet. We need to do this. That the file size are what they're expected.
Yeah.
That, but
I had one last question.
Yeah.
But I don't want to go first. In terms of when you submit your data, do you have options to keep it? I don't want to say secret, but maybe just keep it for yourself and share it with Fabian and
Yeah.
not make it publicly accessible.
Yes. So again, this is one of the last piece that we're making sure that it works as expected. So we haven't put any unpublished data on there yet. But so Gen 3 data commons, so they've got a lot of data commons. They actually have some stuff that's human as well. So there is user level control. So we can say these two people are part of this project. Everything's at the project level. So anything under a project. So it could have multiple studies. So we haven't been able to control for a specific study. So that's one thing we have to consider. But we could say these two people should have access to this project. And we can change those permissions later on. Say, okay, now it's ready to be published. Let's change those permissions to open. Or maybe we change those to just everyone at URI or everyone that's part of Superfund that we have in the list. We still have to add users, so we can't just, so we have open and we have to type in every single user, but it's not that hard. But yeah, we can control access. Yeah. Yeah.
So I noticed that AI has gotten pretty good in extracting information from PDF, text,
Yeah.
papers, and manuscripts. Is there any plan that you maybe at some point just have to upload a paper and it will extract all the information that is in the paper and you just have to add the information that is not explicitly available in the paper?
Yeah, that would be nice. And actually, we've kind of talked about that already in some tools that are available already for doing that. One of the things we're looking at is doing that retrospectively for older studies. We've played around with some tools for that. They're okay. They're good. Definitely found that if you pay for, the newer model, they do better. But that would be great in the future, right? You not even your publication, but you write up your methods and you say, okay, populate my template, and we'd love to do, okay, so it populates the template, puts in what it can find and says, okay, now fill in the gaps. These are the things that you're missing or that we might need.
That would be good to approve the paper then.
Yeah, exactly. Yeah, exactly. Yeah.
Maybe I should report.
Yeah.
Those.
Yeah.
Which we usually-
Yeah, that was I mean, again, it wasn't surprising. It was concerning, but also, like I said, it's in the papers, which was like, so I don't want to knock those authors at all. Like, I think they just didn't think of it when they were submitting, but it is a little scary that it's not there.
Yeah, you want to be able to trust it though, because like in the early days of ChatGPT, I thought, okay, I was interested in converting, say, PFAS compound names into chemical codes. And so I said, can you give me the INCHI code for, and I gave it a list of PFAS compounds. And sure enough, it spit out a list of codes. They were all wrong.
Yeah.
But I mean, that was
It's
early
better.
models.
It's gotten better. You always have
Yeah.
to double check, obviously.
Yeah.
But that's-
So it's
Especially, I think when you upload your own paper where you kind of know what you've done,
yeah, yeah.
would save about.
But yeah, I mean, that's something, because I'm starting to collect all the Steve publications in a database. And there's, and one of the one of the benefits of doing that is, you know, I would like to be able to just have it spit out some recommended terminology for describing data sets or what PFAS compounds were used in this particular study. And I think those things would help sort of as a nudge to make it easier to describing the data.
Yeah.
So we'll see how that works. I mean, you know, it's not at the top of my list of things to do, but you know, it won't be that hard once I have all the PDFs in one place.
Yeah. One of the other things we found as well that we kind of incorporated, so you'll see in those templates, we want both like the chemical ID and the chemical name, because people are used to looking at the chemical name. Like if you go in a database and search for PFAS, you wouldn't know to look for DTX 05, you know, whatever.
Mm.
And so what we do as well is we say, okay, well, does the DTX ID match the name from CompTox dashboard? You say, oh yeah, they match, it's probably right. We also do so. When I first started doing this, you know, because we're so used to working with mice, I started working with a group that did rat. So I would put a rat strain and put organism as mouse by accident. Like, oh, hold on, we can't do that, right? That's wrong. So we're checking, okay, is the strain really a mouse or is it really a rat? So we do have some checks like that to make sure that, is it consistent, right? Is it what we expect? We can't, I mean, we don't check everything. So like we could put normal ranges for what you would expect for temperature, right? Things like that, or humidity shouldn't be more than 100%. See that, but yeah, we try to build in those checks along the way, and when we find some, we add them in. But sometimes some of them surprise us. So.
Data storing is expensive, so what happens if funding runs out? And
Yes, so data storage is probably the most challenging because you'll see we focus mostly on like body weights and things like that right now because they're small and easy to store. Everything right now is on an S3 bucket, which is kind of the most expensive you could do. The nice thing, so Gen 3, again, they're a phenomenal team, and they've thought about this. And so, for example, at MSU, they're building an internal, they call it a Ceph storage, but it works like S3, where it's going to be a lot cheaper. It's going to be on campus, where we can link our accounts, so our data sets go onto that storage. And if another institute has their own, they could go there as well, or they can use an S3 bucket. And then the other thing, one of the main reasons we connect to these external databases like the gene expression data, right? These are big files. And NIH is already paying to store them. So can we just link it to where they're storing it? And that's what we're trying to do for some of these. For mass spec, it's been harder because Metabolomic Workbench doesn't point you to an individual sample. They'll link you to a whole project. And so you can't just download one file at a time. Whether that's better or not, I don't know. I guess I'd have to ask mass spec experts, right? Do you probably want the blanks to come with it, right? And things like that. But yeah, so it's a challenge, and yeah, it's gonna cost money. I probably didn't put my email, but anyone's welcome to email me at any point as well. I can definitely. You can share it if you want to, and yeah.
Yes, and if you could share your slides, that would be great.
Yeah, absolutely.
You can just share it with me and
Yeah, I'll add in my email into
Yeah.
it because I just realized I put in there, and then I'll do that.
Nice. Yeah, thanks a lot.
Yeah, great.
For me, it was great to see a use case. So talking about fair data, I guess
Yeah.
it's kind of.
Yeah, we really started with simple. I mean, it's hard to show a use case with like heavy AI, machine learning and all that because we just don't have enough data in there yet.
Yeah, but you started with. I mean, you're kind of doing it for a use case, right?
Yeah, exactly.
So unfortunately, we don't have lunch in terms of food. But we have the room, so if you want to stay.
If you wanted to
It's
go
fine.
and find food and come,
Or yeah, that works too.
there's a little bit downstairs and then there's the cafeteria.
Just wanted to
That way.
quickly say hi, as well.
Yeah.
Fabian.
Yeah, nice to meet you.
Assistant professor.
I think we'll talk later.
Yeah, exactly, yeah.
Yeah.
So are you getting any tour also of the College of Pharmacy?
If you're giving it, then
I mean,
yes, he's
that's
getting.
probably instead of being here the whole day, maybe not too bad to go. It's like 2 minutes. So I can show you the building, and maybe a lab or two. What time was it?
Meeting with Melanie at 1:30.
4:30?
No, we have a meeting at 3:00. So I think it was 3:15.
3:15,
Yes.
okay. So I'll pick you up at 3:15 and small here. Okay. See you later.
Thanks for coming.
Yeah, sure. It was very interesting. Thank you.
Thanks. Doing Gen Chem, right?
Then have lab, yeah.
And is it like the Gen Chem or Gen Chem for non-scientists?
Oh, okay, good. Yeah.
The Gen Chem for non-majors, so most of them are STEM majors, but they're not chem majors.
When I did that, it was like all like a bunch